today I think it's going to be a very clear beginning because as I wrote to you in the email when I contacted you first, the reason I wanted to talk to you is because you're senior author on one of my favorite papers. And so I'm referring to your science paper, which is one of the two papers we're going to talk about more today. But I thought it might be nice to kind of say why, what I like about the paper so much as a kind of introduction to the topic. So there's kind of three separate reasons in a way for why I think it's a really cool paper. The first is that it offers a kind of first glimpse at what a biomarker for Alzheimer's disorder might be before it's too late, basically. Because as far as I understand right now, you are diagnosed. You're diagnosed with Alzheimer's disorder, and by that point, it's basically too late. There's nothing you can do about it. But with this study, it seems like this might offer some hope and a suggestion for how this might be achievable. And I think that's a huge step forwards. The second thing for me is also, I work with neuroscience with humans and using fMRI. And I think there's always this, at least for me, this like question of like, does the fMRI signal really measure something useful or not? And, because it's so indirect and all that kind of stuff. But then, I don't know, every time I have these thoughts, I think, well, it seems like there might be something there with your study where it seems to actually measure something that's real and that, has some real world implications that, are more than just correlating some activity with some task. And the third reason is that I like that it has this like main story, as in like the title is Reduced Grid Cell Performance in Patients, or not patients, but in not patients yet. That's the cool thing. But then there's all these like lots of, I feel like more than in other papers, lots of additional analyses that really provide a much more nuanced picture about, okay, so you have this reduction in the activity, but the behavior or they don't seem to be worse at the task, but they're compensating for it. And this is explained by this. And then you have like kind of multiple things all in one paper. So I thought, that's why I think it's one of my favorite papers.
Yeah,
that's why I want to talk about it.
Glad to hear about that. Glad to hear that.
Yeah. So maybe as a very basic kind of starting point, how did the project get started? I think you did, I'm not very familiar with the other stuff you've done. It seemed to be more related to memory or something. So how kind of did you go from oscillations and memory to this more kind of slightly more applied work?
Yeah, thanks. So that's a good question. So at the time when we started working at the paper, which was in, I guess, like 2012, 2013, and then of course it took a while before it was published. That was a point in time when the new German Center for Neurodegenerative Diseases started in Bonn. So it's called the DZNE in German, or in English, it's the German Center for Neurodegenerative Diseases. So it's like both a research and a clinical treatment center related to Alzheimer's and other neurodegenerative diseases. And it was just established probably in 2010, 2011. And I happened to be in Bonn at that time at the Department of Epileptology, where I mainly did memory research, as you mentioned. So short-term memory, long-term memory, memory consolidation. And then I found this idea about this center and the plans very intriguing. And I actually applied to become a PI in a junior research group at that center about memory dysfunctions in neurodegenerative diseases, so in particular Alzheimer's disease. And then I thought about which approach would be most useful to understand memory and memory changes and memory pathology in Alzheimer's disease. And the first decision was to focus on very early disease stages for several reasons. So first, because as you mentioned, Alzheimer's disease is developing very slowly, so across several decades, so where the symptoms only become apparent at a relatively large stage. And so currently there's still no really effective treatment for AD, despite many drug trials. And as you mentioned, this is probably because the treatment only starts when the patients have advanced or at least symptomatic Alzheimer's disease. And when you look at the brains of these patients, they are very strongly destroyed already to a large degree. And I just thought it's quite unlikely that in the near future any possible treatment will be successful at that stage. So basically you need to look earlier. And this slow progression is both a good and a bad thing. So the bad thing, of course, is that the disease starts so early even. But the good thing is that if it is possible to find either some biomarker or some cognitive marker that is able to find these very early disease stages, then you may identify patients or healthy participants who are already in a treatable stage. So this was the first decision, not to study patients, but really healthy participants with a risk factor. And then as a risk factor, we focused on a specific genotype, the ApoE genotype or apolipoprotein E. And this is the single one genetic alteration, mutation, polymorphism, however you want to call it, that is explaining the risk for Alzheimer's disease more than any other single gene. As a side note, it should be mentioned that there's other kinds of Alzheimer's disease, so the familiar type of Alzheimer's disease, where there's also genetic mutations that, however, lead with a penetrance of almost 100% to Alzheimer's disease. So then it's not a risk factor, but really a gene that causes the disease. But this is a very rare case, and I found it more rewarding to focus on the more common form of Alzheimer's disease.
I have one quick question about the patients here. Is that, so in your paper you mentioned that there's a 10-fold increased risk of developing Alzheimer's disorder if you have both of these alleles? But I don't know what the baseline is. So is it I guess this is already maybe a fairly technical question, but my question was what I was wondering when I read that was, how likely actually are they in absolute terms to develop Alzheimer's disorders, and therefore how likely are you actually to have people in your study who
So I would need to look up the numbers for Alzheimer's disease, but in the general population the risk of getting Alzheimer's disease changes with age. So at an age of about 65 it's relatively low, it's a few percent. At an age of 75 it's already above 10%, and at 85 it's, I would guess, roughly at about 20%, but I would need to look up these numbers.
Okay,
so it's like a 3-fold increase is clinically relevant. And this is also one of the reasons why when we recruited the participants, the entire study would be completely double-blind. So we would not look up whether a particular person whom we scanned was a risk carrier or not. So we couldn't tell. We only looked that up after all the data had been collected and had been fully pseudonymized. And also there were some participants who asked us whether we could tell them. And we had a long discussion with the IRB in Bonn at that time. And the Institutional Review Board told us that we would not be allowed, even if the participant would be interested, to look that up, because this is information that can be psychologically very distressing, because you know that you have a clinically relevant risk factor for Alzheimer's disease, which is currently not really treatable. So that's why it was fully double-blind.
But wait, just a quick question here again, because what I assumed what happened is that I don't know, because somehow I assumed the recruitment of these participants worked something like, I don't know, you had people with Alzheimer's, and because it's a genetic thing, you can recruit the children or something like that. So I assumed they already knew this, but this was just general population, and then you gave them a DNA sampling?
These were just students. So basically what we did was to go to the big lectures at Bonn University. So there's some lectures with hundreds of students. And then I asked the faculty member who was giving the lecture whether I could have a 2-minute slot at the end of the lecture and tell about the study, and ask whether there would be some volunteers who would be interested in potentially participating. And then afterwards we were standing outside with some students and colleagues outside of the lecture hall and would genotype as many participants as possible. And then from this large database of more than 1,000 participants, we randomly selected risk carriers and non-risk carriers and invited them. But when a given person arrived at the lab, we wouldn't know whether this was a risk carrier or not.
Okay, yeah.
So this was like the basic setup. So we decided to look at young participants, and we decided to look at ApoE for risk carriers. And looking at young participants also had the benefit that we could do this relatively long and demanding fMRI study. So this would not be possible with an Alzheimer's patient. It probably also wouldn't be possible with someone suffering from mild cognitive impairment. And maybe it would be possible, but it would be very demanding and difficult to do, even in the average healthy 70 years old, because for elderly people, it's more difficult to lie still in the scanner without moving a few millimeters for more than an hour and doing the virtual reality paradigm. I mean, just because of like back problems and so on. So we could do this like relatively complex paradigm in young participants. And then we thought, which paradigm should we do and which area should we target? And we relatively early decided to focus on the entorhinal cortex, which is one of the brain areas that is affected earliest in Alzheimer's disease. There's some neuropathological studies that suggest that it's even the first area to be affected. And the entorhinal cortex is an input zone to the hippocampus, and whereas the hippocampus is extremely well characterized, it's maybe the best characterized brain region in both rodents and in humans. This is not necessarily the case for the entorhinal cortex, in particular with regard to memory. There's many studies on the division of labor between the hippocampus and the neocortex, and also between the hippocampus and another adjacent area, the perirhinal cortex, suggesting that the hippocampus is more doing relational memory, associative memory, recollection-based memory, and the perirhinal cortex is doing item-based memory, so other kinds of memory. But the role of the entorhinal cortex for memory is not really that well known. So if you look in PubMed, you find that the number of studies that look at memory in humans, specifically in the entorhinal cortex, is very low. There's a few studies, but it's relatively few. So then we thought of alternatives and so other cognitive processes. And at that time, one of the most investigated research areas with regard to medial temporal lobe were the grid cells that were just discovered in 2004, 2005 in the Mosers lab. And the grid cells are a particular type of cells that is probably putatively relevant for spatial navigation, and that fires in a very specific manner. So like a single grid cell does not fire at one location when you walk around in a room or when the rodent runs around in an environment, but fires in a very repetitive pattern across the environment, and therefore has been suggested to be a neural signature or provide a neural metric for distance estimation and path integration. I guess we come back to that a little bit later. And when we were interested at the time when we planned the study, there was just one publication from Neil Burgess' group with Christian Döller as a first author that was published in Nature in 2010. And that suggested that the grid cells could be studied non-invasively using functional MRI. So I knew Christian from previous meetings and contacted him and asked him whether he would be interested in sharing his paradigm to apply it in the genotype participants and test whether the genotype participants with a genetic risk for Alzheimer's disease would show any dysfunction at the level of this network fMRI pattern of grid cells in entorhinal cortex. And he agreed and sent us the paradigm, and then we scanned the risk carriers and non-risk carriers. So that was basically how the study developed.
That's really interesting. Somehow, yeah, it's fun. I never realized that there wasn't much research on memory in the entorhinal cortex. I mean, I guess I was never really particularly interested in memory per se. So I've had I did psychology and cognitive neuroscience in my bachelor's and master's. So, I've heard about these things, but it never occurred to me. Yeah, it's right. The memory research is always about the hippocampus. It's never about entorhinal cortex. And for me, just because I, in particular, I did my master's at UCL and took Neil Burgess' course with Casper Barry. They taught a course on neurocomputations. So that was all about memory and the hippocampus and entorhinal cortex and grid cells and places like. And it never occurred to me that it's right. The entorhinal cortex was only really mentioned in relation to grid cells.
Yeah.
I didn't realize it was that specific. Yeah.
I think it's still an an area for research. So I think it would still be interesting to try to elucidate and investigate the function specifically of the entorhinal cortex for memory. It's not an area that I'm currently working on, but I think it's still a bit of an open question.
Yeah, and I talked to Kate Jeffrey on the podcast, also a little bit about kind of how this was discovered, because she recorded entorhinal cortex grid cells in the 90s, early mid 90s, but didn't realize she was doing that because the arena wasn't big enough. Basically, it wasn't enough to see the pattern. And I think she also said it was the obvious place to look at where play cells get their firing from, just because it's one synapse away from it. But yeah, that's it. It's just basically you just go well, where does it get its signal from? Let's look at that place. But okay. I never realized that entorhinal cortex wasn't as well known as the hippocampus, but now that you say it, it's kind of obvious. Yeah. Do you maybe want to outline then, I guess we've been kind of talking about the study without actually saying what you did and found. Yeah, just a brief summary of that.
Yeah, so the participants conducted a spatial navigation study while they were lying in a scanner, so in a virtual reality paradigm, which was adopted from the previous study of Christian Döller in 2010. So in this task, the participant had to learn the locations of different objects in the environment and try to remember the locations and then move to the locations. So specifically in each given trial, they saw one object, which were just everyday objects like a clock, or we had an eggplant, a baby bottle, and so on. So all kinds of objects. And there were 8 objects in total, and each of the objects was initially shown at one of the locations in the environment, and the participant was asked to go there, to move there with some button presses on a keypad. So then in each trial, they saw one of the objects, so let's say the eggplant, and they moved to a location in the arena, in the virtual arena, where they believed the eggplant was actually located. And then they pressed a button. And at the beginning, with these 8 objects, it's terribly complicated. So the participants essentially perform a chains. So they just place it somewhere. But then, after placing it, they see the correct location in each trial. So they can then go to the correct location of the eggplant and re-encode or again learn where the eggplant belongs. And then in the next trial, there was another object and so on. So across the entire experiment, which took more than an hour, they saw each object more than 100 times. So you can nicely see how the performance improves for the objects, whereas at the beginning, the participants are very bad. They get better and better across the paradigm. So what this gives you is a measure of spatial memory, which is the inverse of the drop error. So the further away in a given trial the participant drop an object, the larger the drop error and the worse the memory. And this improves across the paradigm. At the same time, what you can record is how the participants are moving in the arena, and in particular in which direction they are moving. And then with a very elegant analysis, you can test whether there's higher activity in the higher bold responses in the entorhinal cortex during movements in one of 6 different preferred directions, which refer to the main orientations of the grid cells, as compared to the directions in between. So basically what you do is identify a hexadirectional modulation of the bold response in the entorhinal cortex as a function of movement direction. So it's a bit of a complicated analysis, but it has been replicated many times now and seems to be quite a robust finding.
Yeah, I haven't mentioned it yet, but I always put the references for the papers we discuss in the description of the episode. So if you want to know more about that, I guess the best place is probably just to read the Döller paper from 2010.
Absolutely, yeah. So then our main hypothesis was that, first of all, we could replicate the finding from Christian Döller because our 2015 paper was the first to try again their analysis algorithm. And then second, we hypothesized that the grid cell-like representations were reduced in the entorhinal cortex. So what we did was to record participants, and then after maybe 20, 30 participants, we said, well, we are still not finished with the recordings, but we would like to know how it's going and whether in the recordings so far we see any grid cell-like representations. And so we applied the analysis that Christian Döller did and found that there were no grid cell-like representation in entorhinal cortex in the first 30 participants, which were like a random mixture of genetic risk carriers and non-risk carriers. And we said, oh, maybe it's just too little. So we kept on recordings. And of course, we had a target number. We wanted to scan 40 risk carriers and 40 non-risk carriers. But I think it's good to do some quality controls also along the way. So we recorded more participants. And when we were at about 50 or 60, we again ran the analysis and again didn't find any significant hexadirectional modulation in entorhinal cortex.
And briefly, if I remember correctly, the analysis is also you look for each participant whether there is this encoding, right? So it's not like you have like one overall analysis that you have for each participant. You didn't find it, basically, right?
Exactly.
That's what you're saying.
Yeah,
okay.
Exactly. So we thought, maybe we will just find that the wonderful Nature paper is not replicable. So this will be an intense discussion with Christian Döller when we need to tell him that we just cannot replicate his results.
Yeah, but Christian, we need to have a word.
But maybe if we want to be very optimistic, it could just be that there are grid-like representations in the non-risk carriers and just no grid-like representations in the risk carriers, and therefore when we lump them all together, we cannot find the pattern, right? Because we hadn't looked at the different genotypes yet. So we just completed the study, and after we had 80 participants and needed to exclude a few, but had more than 35 in each group, we separated the risk carriers and the non-risk carriers, and then indeed found, a bit to our surprise, that our hypothesis was absolutely confirmed. So that the non-risk carriers showed robust and clear grid-like representations, so we could replicate Christian's previous findings, whereas the risk carriers did not show grid cell-like representations. So they were massively reduced to the degree that they could not be recorded, that they could not be found at this network level measure of grid cell-like representations. So this was the first and maybe the main finding in the paper, that in the participants who had an average age of 22 years, so were really young university students with no apparent neuropsychological deficits, but if they had a risk factor for Alzheimer's disease, so a higher risk of developing Alzheimer's dementia in their 60s, 70s, or 80s, already at an age of 22, they showed this massive reduction of the functionality of grid cells at this network level. So that was a bit astonishing and also disturbing, maybe, that in such young participants you already find clear evidence for reduction of the functional grid-like representations.
Yeah, I'd like to ask about get to the other findings in a second. Before I have one question, which is something that just occurred to me, is that did you ever consider not doing the study because it was a bit too risky? I mean, it kind of seems like the Nature paper had never been replicated, as you said, and now you're doing this. I'm assuming you have to sequence the genome of the participants, and then you have to scan them in a long fMRI. So it sounds like a lot of effort for something that, is just this. And I think in Christian Döller's paper also, it's one experiment, right? I mean, one with humans, and then they have like a other side thing with rats. But so basically like one study found this, and now you're putting up a pretty large study. Yeah, I was just curious, like it's, yeah.
It was definitely a high risk, high gain project. So, I mean, it could have been that either we wouldn't be able to replicate the initial finding, or it could have been that there would not be any difference between the risk carriers and the non-risk carriers. It was a risky study, but yeah, we were lucky to find some interesting results.
Okay. So as you said, big difference in grid cell-like representation, but obviously, as we mentioned earlier, people don't develop symptoms until quite a bit longer. So maybe you can link that to the next finding here.
Yes. So the next finding was actually a lack of a finding. So when we looked at the performance, so the drop error and how they changed with learning, there was absolutely no evidence for any difference between the risk carriers and the non-risk carriers. So when we plot the curves of the drop error as a function of trials, it drops dramatically. At the beginning, it's very high. At the end, it's very low. but it exactly overlaps for the risk carriers and the non-risk carriers. And we also looked for a number of other markers, like how rapidly the participants moved, the number of trials that they could do, and so on. And there was really no evidence for any difference. So the most parsimonious explanation of that is, okay, you can use this complex analysis to identify grid-like representations in the entorhinal cortex of humans, but they have no functional relevance whatsoever. It's just some weird epiphenomenon or surrogate marker. So next we tried a little bit harder and conducted a multiple linear regression analysis in which we took multiple different factors into account that may explain together conjointly the inter-individual variance in performance. So in addition to the grid-like representations, we considered the genotype. So whether a participant was an ApoE4 or non-ApoE4 carrier, whether the participant would be male or female, whether the participant would be a little bit older or younger. And then we did a few additional control analyses in which we included some other factors, but these were the main predictors.
Actually, predictors of what? What's the
predictors of individual performance? So of the summed drop error across all trials.
Yep.
And in this multiple linear regression analysis, we indeed found, so first of all, the genotype by itself did not predict performance. The male participants were substantially better than the female participants. We hadn't explicitly looked at that, but this was also a finding that was consistent with some previous studies, like showing better spatial navigation performance in a number of different
It also came out in the Sequest study, right? I think I saw something.
Yes, it's a like a recurring finding. We also found that in a number of follow-up studies. So it just seems to be that the male participants are doing better than females in many navigation experiments. The third finding was that those participants who were a little bit younger were doing better than those who were a little bit older. And interestingly then, the grid-like representations in a given participants did predict significantly the performance. So those participants who had better grid-like representations, more pronounced grid-like representations, also had a better performance. So then we thought, wait a minute, this doesn't really fit together. So on the one hand, we have the genotype effect. So the risk carriers have reduced grid-like representations. The reduced grid-like representations predict performance, but the risk carriers do not perform worse. How is that possible? And then we thought, well, maybe it is because the risk carriers use some compensatory other brain system to do the task. And an obvious candidate is to look at the hippocampus, right? Because of the play cells and the well-known role of the hippocampus for spatial navigation. So we just took an overall sum measure of hippocampal activity during the task-related hippocampal activity, and tested whether this differed depending on whether a participant had more pronounced grid-like representations or reduced grid-like representations. And we found indeed that there was a negative correlation. Participants who had more pronounced grid-like representations had relatively lower hippocampal activity as compared to those who had reduced grid-like representations. So this suggests that you can do the task with two alternative complementary mechanisms or spatial navigational systems, either a system that relies on grid cells in the entorhinal cortex or a system that relies on the hippocampus. So I think this was another fascinating finding because it fits to some previous results suggesting that in very early Alzheimer's disease, there's a relative hyperactivity of the hippocampus. Not only of the hippocampus, but also of other areas of the default mode network, but including the hippocampus. And this may actually be functionally and pathologically relevant because there's evidence from rodents which shows that rodents who have a permanent hyperactivity of a default mode network aggregate more amyloid as the main pathological marker of Alzheimer's disease in this area, which in turn then leads to a stronger progression of the disease. So it may be that when you, at young age, have a reduced grid system, this leads to a compensatory hyperactivity in the hippocampus, which helps you to maintain performance in the short term, but could actually be detrimental and causally contributing at least to the further development of Alzheimer's disease. In humans, this is still more speculative, and it's definitely speculative in these very young participants. but it would be consistent with a number of previous findings. And then the final piece or the final finding from the study was that we looked further whether there was any evidence that the recruitment or reliance of either the entorhinal or the hippocampal navigational system would show up in a slightly different navigational strategy. And what we did was to investigate whether the participants moved either in the center of the arena or at the border of the arena. So you can reach each of the objects via different trajectories, of course, right? And this is independent of the drop arrow. So the drop arrow can be very high or it can be minimal, regardless of your path to this object.
The arena, there's no like objects in the way or something, right? You can just how you get there's pretty free to the participant, right? Yeah.
Exactly. Yeah, maybe I should have described how the arena looks like. So this is basically a circular area with some grass on the floor, which is surrounded by a wall, and in the distance you have some mountains that you can use for directional information. But at any given point in time, the arena is empty, so you only can move around completely freely and then place the objects where you think they belong. So you could either move more along the boundary or straight through the center. And what we found was that the risk carriers were more likely to move along the border of the trajectory than the non-risk carriers. So even though both groups did the task equally well, there was some obvious and statistically very significant difference in the movement pattern in the arena. And this was something that we've just found out through exploration. But interestingly, this finding now has been replicated in a number of other studies, also from other groups. So, for example, in the CE request, they also found that APOE4 carriers, so genetic risk carriers, moved more towards the boundary, which in the CE request consists of the land part of this paradigm, than the non-risk carriers. So it seems to be like a common phenotype of APOE4 carriers that they tend to move closer to boundaries. When we conducted the study, we didn't really know why that was. So we thought, well, if we would have done a rodent experiment, maybe it would have indicated that the risk carriers show higher anxiety, because rodents obviously don't like to move in open arenas, but for humans it's not that obvious. I mean, there's some indications. So, for example, if you go to a restaurant, typically most people prefer to sit at the boundaries or at a like towards the wall than like to expose themselves in the center, but it's obviously not as pronounced. So we just speculated that this may be a reason, but we were not really convinced that it is. And then a few years later, there was a paper coming out from Lisa Giacomo's group at Stanford, where she found that that grid cells actually need encounters with a boundary to stabilize their firing patterns. So when you look at the stability and the accuracy of the firing of grid cells, depending on whether rodents have been far away from any boundary, or probably also other spatial cues, the grid cell firing becomes less stable. So it's probably due to some error accumulation. There's some inaccuracies that develop. Whereas, like shortly after encounter with a boundary, the grid cell firing is very precise. So obviously this didn't really work on the risk carriers, right? Because they moved close to the boundary, but still their grid cell pattern was quite inaccurate. So our current interpretation is that this is actually an attempt to try to stabilize the grid pattern, but it's not effective in the risk carriers.
One thing that I think it actually kind of fits with some of the rodent literature on the development of spatial navigation, because if I remember correctly, these are studies where they do these kind of study with rats when they're just a few days or like 3 weeks or 4 weeks old. And I think there you start off with these boundary cells, and it takes basically at day 14 after birth or something, they have like boundary cells or something like that, and maybe some place cells, but they don't have grid cells yet. That kind of takes longer to do that. So it seems maybe that, it's the same thing. If you don't have this like generalized metric through the grid cells, you kind of have to use whatever's left, in this case, boundaries and
exactly. Yeah, so that's so I think on the one hand one could think that the encounters with the boundary help to stabilize the grid cells just because you get more visual information, but could also indicate that you fall back onto maybe an ontogenetically older navigation system that is more supported by the hippocampus, that is also affected later in Alzheimer's disease, so that putatively is still functional in the young risk carriers, but that shows up in the specific navigation path so that the participants are orienting themselves by moving close to the boundaries.
Yeah, I guess there's two different interpretations of the same thing, right? Either that they're trying to stabilize the grid cells, or they can't use them or use them less, right? It's not like a on-off. One question, I think is kind of my final question about this paper, which is, were the participants, the risk carriers, aware that they were using the boundaries more, or that they were moving closer to boundaries? Is this something that people were aware of?
Not as far as we know.
I guess you didn't even know who they were during the testing, yeah.
Yes, and we also didn't ask them explicitly. I mean, this is something that we are currently exploring in follow-up studies, where we are looking at boundary effects, so the propensity of a given person to move close at a boundary. And also there we are using like questionnaires and ask the participants about their subjective navigation strategies and so on. But in that study, it was basically an exploratory analysis that we did after the data collection had been completed, just to explain how this finding that the participants seem to rely either on one or the other navigation system, so either the grid cell system or the hippocampus, and test whether this was somehow reflected in navigational strategies. But it was very exploratory, and my guess would be that the participants wouldn't notice. But then, I mean, on the other hand, how should they? Because they were either always risk carriers or non-risk carriers, right? So they were not like suddenly in a condition where they moved close to a boundary. So they would need to compare themselves to others and...
Or I guess to their previous selves, right? Supposedly, I guess the idea is that the entorhinal cortex deteriorates throughout their life, right?
Yes, but the question
is... Or is it
really,
that they're born with...
That's really a good question. I mean, the question is when this pathology start, and there's some interesting literature there's some literature from rodents, but in particular there's literature from neuropathology, where the brains of children or adolescents who died not from Alzheimer's disease, but from something, were dissected, and then the entorhinal cortex was studied. And this is mainly work by Brag, who is also did the famous Brag staging for Alzheimer's disease, like a famous neuropathologist. And he found that in some participants, you could even see tau neurodegeneration, which is neurodegeneration that, among other diseases you also find in Alzheimer's disease, can be observed even in children below 10 years. So very young children. And they interestingly also found that the likelihood to see these neuropathological findings is higher in APOE4 carriers. So it may be that even before the age of 22, we would have found some effects.
Okay. So what's the paper with the risk carriers having even in childhood?
Yeah, I believe that is cited in the science paper.
Okay.
Maybe Gebremedin and others. So it's all work from Heiko Brag. So if you cannot find it, just let me know. Then I search for it afterwards.
Okay. Yeah. Great. Yeah, those are kind of the questions. I think we've kind of covered the science article pretty thoroughly already. So maybe we can just move on to the science advances paper, and then kind of have a broader discussion after that. I think maybe one way to summarize the science advances paper in like 2 sentences or whatever is something like, in your science paper, you found that people have reduced grid cell-like activity, but they can compensate with other mechanisms. And in the science advance paper, the question is, what do they do if they can't rely on these mechanisms?
Exactly. So the main idea behind the science advance paper was to develop a paradigm with different conditions. So one condition where you conduct a specific spatial navigation task that you can only solve relying on the entorhinal cortex. So probing the entorhinal cortex with a relatively specific task. So not the spatial learning task that we did before that is known to rely on a variety of different navigational systems. And then on the other hand, use a control condition, or in that case, two control conditions, where subtle dysfunction of the entorhinal cortex can putatively compensated for by recruiting other systems. So basically the main idea is that when you navigate in the real world, typically you use a multitude of different mechanisms. So one mechanism is orienting at allocentric cues. So for example, spatial navigation here at Ruhr University is a very challenging thing. I'm not sure whether you've ever been here. This is a huge campus. It's very confusing. And the simplest way to find your path is to use a landmark. For example, the big Audimax, which you can see from many places, and then you just go there. So this is a relatively simple navigation strategy. I would argue that probably you don't need grid cells for that. You just need to keep this spatial cue in mind, or in your sight, ideally, and go to it. Another strategy then, and that is very different, is landmark-based navigation. So landmark-based navigation is a strategy where you remember specific salient points on a particular trajectory and just remember whether you need to go left or right when you see this point, right? So this is an egocentric navigation strategy, obviously. So when you come from a different direction, you shouldn't go in the same direction.
Yeah. Like the idea being, I go to the petrol station and then turn left. But if you're coming from the other direction,
then you should turn right. Exactly. Yeah. So this is a so-called landmark-based navigation strategy. And then there's a third system, which is called path integration. And path integration means that you basically estimate the distance that you travel and the direction in which you're traveling, and then use this information to find a path. So this strategy can be used. So the advantage of this strategy is that it can be used in the absence of most other sensory cues. So for example, my main example is always when you're in a hotel room and at night you want to go to the bathroom and it's completely dark, so you don't see anything. Then you try to remember in which direction to go, and okay, it's about like 4 meters to go to the door. So you're more or less able to find your way unless there's something standing in your way, right? So, and this is a strategy where basically you estimate the distance to the door and the direction, and you rely on path integration. If you can see the door, you don't need that.
Yeah, or maybe one example, I don't know whether this is true, but it seems like it might be, if you're in prehistoric or before GPS and that kind of stuff, if you were at sea, then you'd probably calculate your speed and with a compass your direction, and then you kind of calculate where you are. Exactly. That kind of idea, right? Yeah. Just, not that conscious.
Yeah. So that's like for a little bit larger distances than hotel rooms. And this is another very good example for path integration. Another example is when you're doing a hike in an unknown environment and, go for a while and then think, well, now it's time to go home. So basically what you need to do is remember how far you went and into which direction, and then you try to find back your initial location.
Yeah, that's what I tried to do last weekend, because I moved to a new place and there's kind of, not mountains is a big word, it's more like a large hill, but through the woods. And I thought, classic cognitive map, I thought I knew a shortcut. I asked someone whether I was going the right direction. I was going the complete opposite direction of where I should have gone. Apparently that didn't work in that case.
I know the situation, and I think it's actually relevant because path integration is a pretty poor navigation strategy. Because when you just estimate a little bit incorrectly, like short distances, then it all adds up and accumulates for longer distances. This is called error accumulation, and is an intrinsic property of path integration. And this is also one reason why, whenever possible, people rely, and also rodents rely, on additional visual cues and use that to correct the path integration error. Similar to the correction of the accuracy of grid cell firing when you encounter walls, right? So I think it's the same mechanism. So we designed a study in which we again invited APOE4 carriers and non-carriers. mainly to do a behavioral study. Then there was also some smaller fMRI study, but the main part of that paper was a behavioral study where we had a typical path integration paradigm. So the participants again navigated through virtual reality, but now they didn't have to remember the locations of objects in the arena. They just started from one location, from an empty basket in that case, and then went to a series of trees to find one tree that contained an apple. That's why we called it the apple game. It's actually Lukas Kunz who is the first author of that study and designed the paradigm who came up with the name. So he should be credited for that. And after finding the apple, what should you do with that? Bring it back to the basket. So basically, it's I would guess like reasonably realistic, naturalistic paradigm, where then with the apple, the participant should find the location of the initial basket. And again, you have a drop error, which is the inaccuracy in a given trial between the distance between the location where the people believe the basket is and the actual location of the basket. So now this is done in 3 different environments. In one environment, this is the most difficult, the participant only see a grassy plain. So they can only look at the visual flow during movement along this arena or space. There's really no boundary and no distal cues to try to orient themselves and estimate the distances. And then there's 2 other conditions, either a condition with a wall that provides some additional information, or a third condition with a local landmark, so which you can also use for orientation. And what we found was that the APOE4 carriers were specifically impaired and only impaired in the task where they had only to rely on the visual cues, whereas in the 2 other conditions, with the landmark and with the boundary, they were not impaired as compared to the controls. So this suggests indeed that when you are in a situation when you can only rely on path integration, so only on the grid cell system, then the APOE4 carriers are worse. But as soon as they can use some compensatory system, they are at par with the controls. This was a relatively large scale study. So this was done in the so-called Apple Game Consortium. So it's a number of groups in Spain, Italy, Germany, and Belgium, who each recruited participants, genotyped them, and then had them done the experiments, and was also not only in young students, but also in I think starting from 20 to 75 years, so across the entire age range, showing that the APOE4 has a very specific detrimental effect on path integration.
Yeah, it's really cool. I mean, it seems to me in a way it's a specification of something you believed or not believed to have found, but like something of an interpretation of the science paper, right? Exactly.
Yes. Yeah.
Does this? I'm wondering. So we just mentioned earlier that the compensation in the science paper through moving closer to boundaries, as we just mentioned. There's at least 2 possible interpretations. One is they use this to anchor the grid cell system to help it, and the other is they just use a different system altogether. Does this paper say anything about that?
Yeah, so the finding is a little bit different, but what we found in the landmark condition in particular is that the risk carriers moved closer to the landmark when they brought the apple back from the tree with apples to the basket. So this suggests that they again stick more to a salient visual cue in order to do the path integration task. So in this condition, they are not worse than the non-risk carriers, but it again suggests that they tend to rely more on additional visual cues.
So I don't know whether you have anything more to say about this study.
No, I think that's the main finding. I mean, I also think it's interesting because it really shows that that there's some very specific cognitive paradigm in which you can see dysfunction of APOE4 carriers, which we didn't find in the previous study. So it may pave the way to very sensitive neuropsychological tests to find early dysfunction.
Yeah, I mean, exactly. So, I mean, I want to slightly broaden out now kind of the discussion. Maybe we can do the application to clinical research in a second. Before that, so one thing I'm really interested in this space is the idea of these abstract cognitive maps, and that the entire system is also used for that. I mean, I talked quite a bit with Jakob about this, Jakob Belmont, and I don't know whether I'm per se going to do research on this, but this is something I'm very interested in, and I'm just curious. So, I mean, maybe to summarize that in like a few seconds, is the idea is that, okay, so this place cell, grid cell, this whole thing we've been talking about, all of that has been initially found, or was initially found, in spatial navigation context. But, well, roughly since your science paper, roughly since that time, there have been quite a few papers that came out that showed that there seems to be some evidence that these grid cells are also involved in processing other dimensions. So we can, if we think of space as having two spatial dimensions, if you're moving in a plane, then there are studies where so that these two dimensions could just be anything else. The Constantinescu paper is with neck length and leg length, which is really hard to say.
I agree.
Of birds in this task. But basically the idea is that maybe this is a much, much more general phenomena where we just happen to find it in space. So I'm curious just if we apply that kind of idea, or maybe let's just assume for a second that it is the case, to your studies. I'm wondering, so one thing I also asked Jakob, and we didn't really have a good answer necessarily for this, is for example, what would a landmark be in this kind of abstract cognitive space? Or maybe I'm asking maybe one question before I should ask another one. I'm not sure really what the question here is. I guess are you planning to do anything like that in that direction, or you're nodding? Yeah, I guess maybe.
Yes. So I found the Constantinescu study very intriguing, and I think it filled in a way like a gap in the theory about the medial temporal lobe, so entorhinal cortex and hippocampus for cognitive functions in general. Because so what you see is that in rodents there's place cells in the hippocampus and grid cells in entorhinal cortex. In humans there's some mixed evidence, one has to say, about the existence of place cells actually. So like using microelectrode recordings in epilepsy patients. Some studies found place cells, other studies did not find place cells. There's some empirical reasons why it's maybe unlikely to find anything like a place cell-like representation in fMRI, because adjacent place cells do not show any topography. So if you average across a larger voxel, probably you cannot decode any specific locations. Although some other studies suggest that actually it may be possible, so it's an ongoing discussion. But there is good evidence now from human single-cell recordings that there's the so-called concept cells in the hippocampus. So which are like the famous Jennifer Aniston cell or Luke Skywalker cells or whatever, which code in a perceptually invariant concepts like particular people. So then this suggests that you have cells in the hippocampus which represent a point in either physical space or conceptual space, whereas you have cells in the entorhinal cortex that provide a distance metric, so that code various different locations in physical space. So does this also occur in concept space? And this is, I think, exactly what the Constantinescu paper suggests, that actually you have these two different types of representations, either like a metric representation of distances, of both physical distances or conceptual distances in your entorhinal cortex, and a very specific representation of individual points in either conceptual space or physical space in the hippocampus. And this also fits the idea to that the hippocampus provides a kind of an index or pointer to representations in the neocortex, which can be very flexibly applied depending on task demands. So what rodents often do in experiments is running around, and also the real world. So it's obvious, or it's quite maybe no surprise, that actually what you find in the hippocampus is representations of specific locations because they're running around. What humans often do is reasoning, making comparisons between concepts. So it may be no surprise that actually what you find is specific representations of individual concepts. So if the epilepsy patients would run around all the time, and if you would be able to record during this period, maybe it would be easier to find also place cells. But the same cell in the hippocampus of an epilepsy patient, which represents a concept, may in other task situations be recruited to serve as an index for specific locations.
I mean, is this then, I guess, is this the secret work you're working on and can't really say too much about? Or is there something concrete planned with, something like this?
Yeah, I mean, this is very much ongoing, but I mean, the main idea is to investigate this navigation in conceptual space. What we are trying is to go a little bit beyond the artificial birds from the Constantinescu study, which are wonderful because they are so well controlled. But on the other hand, of course, they are artificial. So
also, it's physical still. Right, it's still about length, distances, which I find is, in some sense, it's not as abstract as they say it is.
That's right. Yeah. So I think that in any conceptual space where you need to make comparisons between pairs of objects, I would expect to find, we haven't really shown that yet, and we don't have any even preliminary results supporting that, but I would expect and hypothesize to find similar representations of distances between objects, between faces, between whatever stimuli you can think of. And then one hypothesis would be that actually path integration and navigation in this conceptual environment would correspond to comparisons. So for example, when you see a person and a person's face and compare its similarity across the high-dimensional face space, which consists of nose lengths, whether someone has a beard, and so on. Like, just think of all the features that make up faces, and you compare this person's face to another face. Or you compare two different objects. Or maybe you even compare in social space. There's a number of papers about that now. Like two people in terms of their approachability or other social dimensions. So whether you like them, whether you don't like them, and so on. So I guess this navigation and the distance estimation in this conceptual space just probably, it's my hypothesis, would reflect similarity judgments in any space.
Mm-hmm. Yeah, it's really interesting. I mean, I'm particularly interested in this kind of social aspects because I do kind of social interactions and that kind of stuff. There's nothing like. Basically, I have like one fMRI study left in my PhD, and we're not sure whether we're going to do grid cells with that or not with the social stuff.
You should.
Yeah, that's I mean, the problem is kind of that for the stuff I have studied to make it like fit also into the stuff I've done before. No one has done this with, so it would, or rather kind of study one would be to establish whether this is a thing in this way I'm doing it at all, and step two would be what I'm actually interested in doing. But that's kind of what I will be figuring out in the next few months. Basically, the thing that I also discussed with Jakob was this question of how far can you translate the physical findings into these abstract conceptual stuff, or to what extent something is specific to space. But you'd think that there is something like landmark-based navigation in the sense of, in this case, as you mentioned, comparing it to something you already know. Or
yeah, I mean, I agree. It's still a bit of an open question what a landmark in conceptual space is, what boundaries in conceptual space are, and so on.
Yeah.
So I guess one possibility for a landmark would be like something like a prototype, right? Like a prototypical representation of, let's say, a particular object. Right, so when you close your eyes and think of let's say a beer, it's getting late, and then something comes into your mind. So this is a prototype of a beer, and then when you're comparing two other beers, you may compare them with regard to your prototype, right? So so when you see some bottle and are asked to judge how typical, for example, it is of a beer bottle, then you compare it to your prototype of a beer. So this may be something like a landmark-based navigation. And boundaries, I guess, may reflect the boundaries of like between concepts, which are maybe not very easy to define, but there's probably some boundaries where some objects where you wonder whether this is a glass or a cup, for example, right? So there's a prototypical glass, there's a prototypical cup, and there's like some things which are somewhere in between at the boundaries between them.
Like a cup made out of glass or something.
Yeah, exactly.
Yeah. Yeah, I mean, yeah, I guess we're not going to get a definitive answer today, but
No, I'm
it's something
sorry.
I find really interesting.
Yeah.
But okay, I guess we've got maybe like 10 minutes left, so shall we maybe move towards the kind of clinical applications? Whilst I was, well, at the same time that I was inviting you onto the podcast, I was by pure coincidence reading Brave New World Revisited by Aldous Huxley, and there was a quote in there that happened to fit, I think, perfectly. I guess I'll just read the quote. The quote is, "Pure science does not remain pure indefinitely. Sooner or later, it is apt to turn into applied science and finally into technology." And it seems to me, I mean, this is also kind of what I found so exciting about your paper. That so we had the basic science with John O'Keefe just, putting a more or less randomly an electrode into hippocampus and letting rats run around, and then 40 years later, we have the beginning of clinical applications, right? So I guess maybe as a very broad question, so how far are we away from this being in any way clinically usable?
So, very good question, of course. So,
predicting the future is, of course, always easy.
So one caveat, of course, is that it's, I think, quite unlikely that you can, or what you cannot do is just take the paradigm, the spatial navigation paradigm, also the Apple game, and test 60 or 70 years old in a scanner in order to see whether they have some alterations of their grid pattern. For practical reasons, this is not really feasible. And what is still an open question is, what we've seen is that there are differences in grid cell-like representations and also navigational patterns and hippocampal activation between risk carriers, ApoE risk carriers, and non-risk carriers. But then on the other hand, like the easier approach to find the difference between risk carriers and non-risk carriers is to genotype them and not do an fMRI study. So in other words, it's currently not clear to which degree these findings of reduced grid cell-like representations or altered representational strategies provide any additional clinically useful information to predict Alzheimer's development or Alzheimer's pathology in a given participant. So what I could imagine, what is much more feasible, of course, than an fMRI study in participants at risk for Alzheimer's disease or with like very early stages of Alzheimer's disease, is to do a cognitive paradigm and to try to develop a cognitive paradigm, maybe something like the C-Hero quest, to find subtle alterations in early disease stages. Then, of course, many validation steps are necessary. So one, of course, is a longitudinal study. So test whether in a 60-year-old who has subtle dysfunction or even only a change in navigational strategies in these paradigms, whether this participant is more likely to develop Alzheimer's disease than another person who doesn't show these deficits or these alterations in strategies, and whether these early changes predict the development of Alzheimer's disease more than established clinical markers, including ApoE4. So this is just an empirical study, an empirical question. Obviously, longitudinal studies take a time and are time-consuming and expensive to conduct. I would love to do a similar study in the future. I think it would be a necessary next step. I know that there's ongoing studies also with the C-Hero quest paradigm on this question. So it's, I think in the end, it's also an open question which paradigm is best suited, is most sensitive, most robust. But I think there will be development on that. Another question, of course, then is whether the changes in navigational strategy are actually reflecting Alzheimer's pathology. In order to address that, one would need to relate them to other biomarkers of Alzheimer's disease. So CSF would be one obvious biomarker which you cannot really obtain from healthy participants. Another one is blood markers of amyloid, which have been established very recently. And then, of course, what is very exciting is the new possibility to do amyloid and tau PET imaging, right? Where you do not only measure the overall amount of amyloid or tau in the brain, but even their regional distribution. So you can distinguish between, let's say, hippocampal or extrahippocampal, maybe even entorhinal tau pathology. Of course, the spatial resolution of PET is somewhat limited, but there's now the second generation of tau PET markers out, and there definitely will be further developments.
What I find really exciting about this is kind of not the PET-based scanning or whatever, because, who has that scanners, right? That's very, very few people can do that kind of scanning per year, very few patients. Whereas what's much more exciting, as you mentioned, for example, the See Hero Quest is a game. For those who don't know, it's like a smartphone-based game. You can just download it in the App Store or whatever. And there's of course some like ethical problems with just anyone who uses boundaries a lot to just tell them, hey, you're using boundaries a lot, you might want to see a doctor. That's probably not how it's going to work. But I really like the idea of, you're at the doctor's and they say, hey, just play this smartphone game for like 10 minutes while you're waiting. And then maybe that already tells you something, right? Yeah,
exactly. So I think this is a likely development. And for me, what is so fascinating about this research direction is that you can draw a line between, on the one hand, very applicable methods like apps or behavioral paradigms on a laptop, even, which are relatively easy to conduct in larger populations. So in the Apple game study, we had more than 300 participants who conducted the paradigm on the one hand, and then the possibility to do the same experiment in the scanner and look at the bold responses. Then what we also did, what we didn't talk about, is run the same paradigm in intracranial EEG in epilepsy patients, so that you could look at the electrophysiological pattern in entorhinal cortex and hippocampus. You could even look at the single cell pattern in some epilepsy patients who have microwires implanted. And then you can see whether any changes in a given person are related to tau pathology or amyloid pathology. So this obviously will not be done in a single study. So you will not have like an epilepsy patient who afterwards does a tau PET, but
it's a big study here.
But basically you can combine these different angles and these different pieces of evidence and then try to develop paradigms that are very sensitive and very specific and easy to run, which may allow you to increase the predictability of Alzheimer's disease in a given person. And at the same time, you can validate the same paradigms by looking at grid cell-like representations in fMRI, single-cell activity and intracranial EEG oscillations in epilepsy patients, and amyloid and tau PET and other biomarkers in various individuals.
Mm-hmm. Final question. So if I understand it correctly, you got a consolidated grant from the ELC to work on this, right? On this kind of research area. Can you say kind of that was fairly recently, right? So kind of what's the plan there? What's the rough direction you want to go down with this research?
Yeah, in just one sentence, exactly what I just suggested. So, like trying to understand how behavioral alterations are related across paradigms, how they relate to fMRI, grid cell-like representations, to electrophysiological signatures of grid cell-like representations and grid cells in humans, and to Alzheimer's pathology.
And that's in physical space or also abstract cognitive space?
That's in different spaces, including conceptual space.
Okay, cool. So I don't know whether there's anything else, any final words you want to say. Otherwise, I'll stop recording. I don't have like a formal ending.
I don't know. A few years ago, there was a so-called grid cell meeting in London. I'm not sure whether you participated in that as well, where the different lines of direction of grid cell research came together. So in addition to what we talked about, of course, all the rodent research, the really fascinating research in bats from Nachum Julanowski's lab, and also the modeling. And this was one of the most exciting meetings that year and for multiple years, I think. So what you could really see is that there's a very strongly divergent, but at the same time, very interdisciplinary and methodologically diverse field, which is progressing very rapidly. And I think it's a very active and fascinating research direction. And for anyone who isn't working on grid cells yet or grid cell-like representations, I just recommend to go in that direction.
Okay, cool.